Good afternoon, everyone. For those of you who may not have met in person or online, I'm Rick Lufkin, chair of the Cardinal Gray Society and a proud member of the MIT class of 1968, soon to be celebrating our 55th reunion. Our class is excited to celebrate on campus in Cambridge in June, but I'm also thrilled to connect and learn with you all today online. We have members of the Emma Rogers Society, the Catherine Dexter McCormick Society, and of course, the Cardinal and Gray Society this afternoon. Welcome all. Allow me also to extend a personal welcome to today's lecture from both me and my wife, Ruth, and express our thanks to you for joining us. While it has certainly been a joy to celebrate once again at in-person Cardinal and Gray events over the past few months, I'm grateful that our community has been able to supplement those gatherings with online events like today's that give us a much wider reach. We have more than 300 Cardinal and Gray members from all over the world registered for today's program, testimony both to our faculty speaker and the enduring bond that we all share as part of the global MIT community. Ruth and I look forward to many more years with Cardinal and Gray in your company, both in person and online. And now allow me to introduce our guest speaker, Nikta Fakhri, the Thomas D. and Virginia W. Cabot Career Development Associate Professor of Physics. Professor Fakhri works in the Physics of Living Matter Systems Group. The title of her talk is Broken Symmetries in Living Matter. Her research focuses on the processes in living and non-living matter that create non-equilibrium materials, and her lab is interested in developing tools for discovering how spatiotemporal patterns arise in active biological materials. Professor Fakhri grew up in Tehran, Iran, and earned a BS in Chemical and Petroleum Engineering from Tehran's Sharif University of Technology and a PhD in Chemical and Biomolecular Engineering from Rice University. She joined the MIT Department of Physics in 2015. Please join me in welcoming Professor Nikta Fakhri. Professor Fakhri, you're on.
Okay. So thank you very much, Rick, for the kind introduction. And thank you all for being here. It's truly an honor to be invited to give this talk to the society. And I'm very excited to share with you how my group has been pushing the boundaries of non-equilibrium physics in living matter and how actions within the walls of a living organism can be accounted for by physics and formulated in mathematical language. Like many biophysicists, let me start by a question that many of us have been pondered upon. And as a physicist, I'm fascinated by the fact that there is something different about being alive. And we would like to know what this is in the same way that we know what it is for a collection of atoms to be solid and for a collection of electrons to be superconducting. When we look around more or less immediately, we can identify things which are alive. And the criteria that we use in making this discrimination between animate and inanimate matter has nothing to do with the individual components, such as DNA or proteins. Being alive is a macroscopic state, while things like DNA and proteins are the components of the microscopic mechanisms by which this state is generated and maintained. To drive this point home, let me talk about this beautiful video that you see on the slide here. This is an egg cell or oocyte of a sea star. It's a model system that we use in my group. And note that this is a very large cell. It's about 200 micron in diameter. Now, what you see in this video is self-organized patterns of signaling proteins that orchestrate a very important process very early in development, namely cell division. And of course, the ability to divide is a hallmark of life as we know it. Now, we cannot see individual proteins. As you may remember, typical protein sizes are on the order of nanometer compared to the size of this cell. And the majority of us may not even know what exactly they are. But we all can appreciate one thing just watching this video, And that's the fact that long wavelength patterns and waves that span the entire surface of the cell propagate from one end of the cell to the other end. They self-organize from one state to the other state. And this happens while the X cell is alive. It's continuously consuming ATP and dissipating energy. Now, over the past many decades, of course, molecular biology has been very successful in identifying the components as well as the microscopic mechanisms. However, I think the intellectual ledger is not yet balanced because the data are outpacing the corresponding conceptual understanding that accounts for the data. In other words, I believe we have made less progress in identifying the parameters that are characteristic of this macroscopic state or life. Let me quote Ernest Everett Just, who's a pioneering biologist, where he says that the direct analysis of the state of being alive must never go below the order of organization, which characterizes life. And if you think about it, this is an approach which is at the heart of physics. Understanding in physics demands that we combine the factual knowledge based on experiments by conceptual knowledge. And one of the fundamental questions in physics is not only discovering the underlying laws in the system, but in understanding and explaining the new laws that emerge when many individual components interact. And to discover these laws, we do not always keep track of all the electrons and protons. We're always looking for important variables, or what we call key degrees of freedom. And once these variables are identified, we combine them into what is called an order parameter, which is a quantification of the emergent phenomena. In equilibrium physics, which is physics of the everyday life, the concept of order parameter has been very powerful, and it plays a crucial role in defining a wide range of phenomena. So let me illustrate this by one very famous example, if you recall from freshman physics, which is the magnetization. So in a magnet, we do not care about the position of each atom. What we care about is this global order parameter, which is the magnetization of the material. Now, asking for the order parameters of the living state is a very hard problem. It's also not a well-defined question. And this is because when equilibrium is given, if you recall from statistical physics or thermodynamics in physics, when we have equilibrium, statistical mechanics frames the behavior and phenomena in terms of minimizing the energy, because we assume that there is no energy that's added or lost from the system of interest. But now if we have a system that's out of equilibrium by necessity, which is life, we can no longer follow an energy optimization principle. And let me quote a famous saying where we say that when we reach equilibrium, we're dead. So we really need to think about what happens when we are out of equilibrium, so we don't really have any optimization principle. So one of the questions is, can we identify general principles that may guide us in developing a physics of life that can reach the same level of predictive power that's the standard as we know it in any other area of physics? And of course, this is a challenge that me and my group have set out to address. And to do so, we develop experimental tools to observe and identify non-equilibrium degrees of freedom. We develop data analysis frameworks to define order parameter fields. And most importantly, we develop theoretical frameworks that have a predictive power. Now, the concepts that I will introduce today are system independent. And I think that's the hope, that's the ultimate goal of a physics approach to living systems. We want to go beyond just the normal molecules and in their interactions. We want to have something that spans beyond a certain scale and system size. So to develop these concepts, however, we need model systems which have rich phenomenology. And to make this clear, let me say we need our hydrogen atom. And if you think about it, hydrogen atom was a quintessential test case for what it meant to account for the behavior of the atom, the nucleus, the coupling between radiation and matter, both Einstein, condensate, and many beyond phenomena that we got to know and understand very well in physical systems. So in my group, we use the O site of a C star. And let me kind of make a case for why this is an interesting model system to choose, because this is actually one of the oldest known developmental biology model systems. I have a reference here that dates back to 1877. And if you think about it, multicellular development starting from this single oocyte, this single large cell, as it has evolved over the past billion years, is truly an accomplishment in the realm of self-organization and information processing. The processes need to be orchestrated in space and time to give us robust outcomes, for instance, cell division. So today, just to illustrate my approach, I'm going to take you on a journey in space and time and showcase some of the broken symmetries that we have identified and the emergent phenomena that we have discovered. And to do so, I'm going to start with the smallest scale, which is at the level of the intracellular scale, within the confinement of a cell. And what I will show you is that how we can quantify time reversal symmetry breaking, or the emergence of the arrow of time using the fluctuations of intracellular organelles, and how we can use tools from information theory and stochastic thermodynamics to identify spatio-temporal scales of dissipation and entropy production, which is a feature of living systems in complex systems. Then I'm going to move up the scale as well as complexity and talk about this beautiful pattern that you saw on my second slide. And I will show you how biochemical signaling proteins self-organize at the cellular scale and how cells use these patterns to perform computational operations. And lastly, I'll tell you about a phenomena that I'm really excited about, and that's my group's recent discovery of a collective phenomena and the subsequent emergent phenomena known as odd elasticity. And I will describe what odd elasticity is, which is beyond what we know about elastic systems, but it actually happens in systems when you have two requirements. One is when you have broken time reversal symmetry, which is a feature of active living systems, but as well as non-reciprocal interactions that defy Newton's third law. And in each of these examples, I will demonstrate how the actions within the walls of this living organism can be accounted for by physics and formulated in mathematical language, making it possible for us to make statements about the phenomena not yet seen, or even measurements not yet made. So let's start what's arguably the most important broken symmetry in living systems, which is time reversal. And to talk about time reversal symmetry breaking, I'm going to start by showing you a video of a developing embryo of a starfish. And this is the first frame that you can see. And let me orient everyone. So in this first frame, you can see many cells, and the cell nuclei is marked in blue. So we have about 64 cells in this first field of view. So I'm going to start playing the video, and we're going to watch this for a bit. So this is just a beautiful video to watch, but I'm sure the majority of you are beginning to realize that there is something unusual about this video. And hopefully sooner and not later, you will recognize the fact that I was actually playing this video backwards. And this highlights a fundamental feature of living systems, which is at the expense of dissipated energy, living systems perform irreversible processes that allow them to propagate and reproduce. And as dictated by the second law of thermodynamics and the fluctuation theorems that accompany that, irreversibility in non-equilibrium processes can be quantified in terms of how much entropy such dynamics produce. this is an absolutely powerful theoretical result. It's one of my favorite mathematical equations at the moment. So I'm going to just walk you through this. So what you see here is that I have written basically an equation that some of you may recall. It looks like a Shannon entropy-like equation. And this result actually shows that there is an exact relationship between the entropy production and the distinguishability of a process from its time reverse, quantified by relative entropy between forward and backward state. So let me just say what exactly this means. So here, what you can see is that the left side of the equation is dissipated work, so it's entropy production. And the right side of the equation is a well-known and powerful concept from information theory, which is called the Kullback-Leibler distance between two probability densities. So this is one probability distribution, the other one is another probability distribution. So if you look at the equation in the bottom, when you look at the Kullback-Leibler distance between two probability densities, we're trying to measure the difficulty of distinguishing whether observed data corresponds to one distribution or the other distribution. In other words, how difficult it is to distinguish the sampling between two densities. Now, if you look at the top, you will see that I've written this equation to measure the difficulty of distinguishing whether my observed data corresponds to those from a forward or backward experiment. So in other words, this relative entropy term can be considered as a quantitative measure of the arrow of time and of the dissipation. So it basically tells us the length of the arrow of time. Now, this is, of course, a theoretical result. It's exact. It holds in any system, quantum, classical. But what we're going to think about is complex systems. So systems that are not just a single molecule doing something, but rather when you have a collection of these molecules together. And what happens is that when you have a system with many, many degrees of freedom, many of the interacting particles, because of the experimental capabilities that we have, often we can only measure one or several of these variables, which means that when we apply this equation to the experimental data, this equation is going to give us a lower bound for the entropy production, not the actual full entropy production. And I'm going to call this lower bound irreversibility. So how irreversible the process is. So this is, of course, it's a powerful theoretical result. The question is, can I apply this to experimental data and measure this thermodynamic arrow of time as it emerges in active and living systems? Now, if you think about it, the video of the developing embryo that I showed you is an unmistakable example of an irreversible process. So it's cell division. You never go back. You never merge two cells together. However, time reversal symmetry breaking actually happens at much smaller scales, and it manifests at system dynamics at much larger scale. So we actually wanted to go to much smaller scales, which is at the level of intracellular scales within a cell itself, and understand whether non-equilibrium activity emerges from the interactions within these structures within the cell. And let me just give you a quick refresher of what kind of structures we have inside the cell. I think one canonical structure that we have is the actomizing cortex. So it's the components of the muscle, but when it's in the cell, it's actually quite disordered. So this is a schematic of this actomizing cortex. It's actually a quasi two-dimensional structure that sits beneath the cell membrane, and it consists of dynamically cross-linked filaments, actin filaments that continuously polymerize and depolymerize. the network is connected, anchored to the cell membrane, and then it's internally activated via the myosin motor proteins. So within a muscle, you have a very ordered structure of this actin and myosin proteins, but within a cell, you actually have a disordered network of these actin filaments, and then the myosin motors, what they do, they connect to the network, they exert some force, and then they release. So if you can imagine, there's a lot of fluctuations within the cell because of the motors attaching and detaching. So the question is, can we look at the fluctuations within the system and try to infer this thermodynamic arrow of time using the data that we can acquire? And of course, because we have to construct an information theory metric, we need a lot of data. We need very long time series data of fluctuations to be able to construct meaningful information. And my group has been developing many tools to be able to achieve this goal. And recently we made a serendipitous discovery, which allowed us to actually develop this tool. And what you're looking at is one of these oocytes, one of these egg cells that we take and we sandwich between two cover slips. And then what you're looking at is the bottom layer of this cell, which means that we are very close to the cell membrane. So we're looking within the actomyosin cortex. And what you are looking at, we do not see the actin and myosin, but we are seeing like a bunch of these granules, this spherical like objects that, as you can see, they're just jiggling around. So this turns out to be a fluorescence microscopy of these granules, which we discovered to emit above 900 nanometer infrared. That's a different story that I will be happy to tell at a different time. But this is a very long wavelength to look at, which means that we don't get any background from the environment. So all we are getting is the signal of these granules, which we can collect with high precision and high spatial temporal resolution. So we can start looking at like where does these fluctuations come about? So why these objects are moving around like this. And you can see we have many of these granules. So the type of data that we can collect is the position of each of these individual particles as a function of time, or what we call trajectories. So now this large position time series data will allow us to quantify irreversibility, which is this information theory metric that we are interested in. So let me show you how the data looks like. So here you can see examples of the trajectories of these particles in time. And what we are going to do is we are going to quantify the velocity of these particles. And to calculate the velocity, we have the position information, so we can use a lag time to quantify velocity. And one thing we did is we actually changed this lag time. So we made it into an adjustable variable. And the reason for that is going to become clear. But what you need to kind of know is that when you increase lag time, what you're doing is you're coarse graining the data. So by varying theta, this lag time, we're generating a collection of observables whose estimated KLD or irreversibility will capture the entropy reduction on scales that are larger than theta, scales that have not been smeared out by coarse graining. Now, so we now have all these trajectories, long time series trajectories. Let's first see if we can understand what we should expect to see in a system like this. And to do so, what I'm going to show you is you can look at a particle, and you can do this analysis where you look at the velocity autocorrelation function. So it's basically you look at one particle and you will see how likely it is to be at a certain distance or a certain location after a certain time. And this is what we have plotted here. So this is the autocorrelation function of these trajectories as a function of correlation time. One thing you see is that, first of all, it starts at 1. This is how we constructed it, but then it becomes negatively correlated at fast time scales. And then, so what does it mean to be negatively correlated? What it means is that you are one of these proparticles, and you get a kick from the environment. And because you're sitting in a mesh network, it's very likely you're in what we call a locally elastic network. So it's very possible that you will just get kicked back to your initial position. So you have this basically negative correlation. And this short time scale dynamics is consistent with being primarily driven by thermal fluctuation. So it's just because we are at room temperature, the experiments are at room temperature. So the thermal fluctuations, just Brownian motion, will cause you to go back to your initial position. But then something happens when you look at much longer and longer time scales. And if you recall, I mentioned that we have these Meissner motors that are sitting also in the environment and also creating fluctuations within the network. And those fluctuations require energy because Meissners need to consume fuel, ATP, to be able to generate that type of fluctuations in the environment. So that's an active form of fluctuation. And in fact, this active form of fluctuation will cause the particles to diffuse away. And that's what basically you see in this function. So the autocorrelation function, the particles are diffusing away when you look at long time scales. So this system then shows two time scales, fast time scales. Basically, the system behaves like being in equilibrium. It's dominated by thermal noise. It's only at time scales that above 1 to 10 seconds, which is the time scale of this Meissner motors, how much time they take to attach to the network and detach to the network for them to actually be able to diffuse away. So this this is what I hope you remember from this slide is that there is this 1 to 10 seconds that a time scale that allows the system to actually behave in a non-equilibrium way, like in not just a thermal way. So now what we can do is, okay, we now understand what the system should behave like. Now let's see if we can construct this KL divergence and see what information we can learn from this. And this was, I'm not going to talk about the efforts that went into developing these algorithms. This was actually done by one of the MIT's undergraduates who was doing a Europe in my group, Gareth Watson. And what he did is he constructed algorithms to be able to take the data and then measure this irreversibility metric, and then basically put everything in this figure, which is an absolutely surprising figure. And I'm going to walk you through this and what we have learned from this. So first of all, what you see we have plotted is this irreversibility metric. So that information theory like term that I mentioned, as a function of the coarse graining variable that I introduced. So how much of the fluctuations we're going to gloss over or just average out. And what you see is the first thing you see is that this plot is, in this plot, irreversibility is a non-monotonic function of the lag time. And it happens to have a peak at about 1 to 10 seconds. So what is this figure is telling us? So this non-monotonic behavior is telling a story that as we probe progressively longer and longer timescales by increasing the lag time, first we capture the reversible thermal motion. So at fast timescales, the irreversibility is small. It means that the system is closer to equilibrium. Then at intermediate times, we have an increase in how irreversible the system appears to be. And then eventually this irreversibility is going down. And that's because we are coarse graining away too much information, which then results in this observed smaller irreversibility. Now, what I would like you to remember from this figure are actually there are two takeaways, and we have also theoretically verified this. So the first thing is that this arrow of time or irreversibility is associated with specific time scales. So if I'm an observer, I can be fooled into believing a process is reversible, just driven thermally, if I watch the system on the wrong time scale. But the second important takeaway from this is that we can infer this active time scale using a completely model independent data-driven metric, which now allows us to actually go in and characterize the dissipation in the system. So there is this notion of the scale and dissipation, which now we have a tool which is fully data-driven, which allows us to go in and now make understanding about the mechanistic phenomena that give rise to dissipation in systems like this. So this is an example of, again, you can take high resolution data and you can use a mathematical modeling to make predictions about the system. So I'm going to kind of wrap up this part of my talk, and I'm looking forward to questions during Q&A, but I'm going to move up the scale and as well as complexity. So at this scale, all we saw was seemingly random fluctuation, yet they require energy. But let's go one scale higher, which is at the scale of the cell, and then try to understand where does these patterns come from and how cells use this type of patterns to perform computational operations. So to talk about this type of patterns, let me start by first kind of introducing chemical waves in general. I think we have all encountered different types of chemical waves in many different fields, including chemistry and even at the climate scale. And these type of waves usually are dissipative structures that emerge in certain type of systems when you have, for instance, in chemical systems, you have reaction and diffusion processes happening at the same time. And in living systems, they tend to be very important because they act as ways of spreading information rapidly to synchronize and coordinate biological events. Again, if you think about it at the scale of a cell, if you only want to rely on diffusion or thermal processes to take you somewhere, you will never get there in time. But when you have this type of waves, they actually spread the information rapidly so that you can synchronize and coordinate events. Now, I'm going to focus on one particular pattern, protein pattern that's actually very crucial for many developmental processes. And this is an example of what these protein patterns can do. So I'm going to play the first video here. And again, this is my favorite model system. It's a starfish egg cell. And by the way, this process is common across all the species. So it's this type of patterns happens across many and any type. It's a well-conserved protein. And what this video is showing is one of the first stages in development where you have this very large cell and it has to divide asymmetrically so that it can get rid of half of its genetic material so that it's afterward it's ready for symmetric cell division. So this asymmetric cell division is called meiosis, and this asymmetric cell division is actually achieved by a certain type of protein, which is known as Rho-GTP. And I'm going to ask you to first focus on the shape deformation that you can see in this video. And you can see that the cell shape is dynamically deformed, but by a surface contraction wave that travels along the membrane from one pole of the cell, which we have marked as VP, to the opposite pole, which we have marked as AP. Now, once this contraction waves gets to AP, a small part of the cell and half of its genetic material, it pinches off. It's called polar body extrusion. And then now the cell is ready for symmetric cell division. Now, if you focus on the green signal, you can see that this contraction wave is in fact induced by signaling protein Rho-GTP, which what it does is it locally triggers actomycin contractility, the same network that I talked about in the first slide. There you have just random activity of myosins, but when you have this signal, it actually tells where and when actin and myosin should get activated so that you get a certain type of contractility that will propagate along the surface of the cell. Now, what we observed, something very intriguing. Of course, as a physicist, the thing you want to do is you want to understand the role of geometry, shape, and everything. So we took these cells and we put them into molds, into PDMS chambers of different shapes. So you can see two examples here. You can have an ellipse, a star, why not? And what you see is that no matter what we did to the shape of the cell, this process happened very robustly, as if the cell knows exactly its precise shape. And that is a very surprising observation, because again, these proteins are very small, about nanometer in size. The cell is very large, about 200 micron in size, and yet the proteins know exactly where they need to be when they need to be somewhere, so that they can perform this process very robustly. So this actually led us to uncover a shape adaptation mechanism that actually arises when you have a hierarchical sequence of protein patterns, or what we call patterns forming patterns. So I'm not going to go into details, but you can identify all the important components of this system, and then you can write down your equations. Well, then describe this whole pattern forming pattern hierarchy. And what I'm going to tell you is basically the main important point from this, which is basically you can have different types of pattern in the cell. So you can have a gradient within the cell, and the gradients is where the shape cell information is encoded. And the way the gradients are decoded is by using a bistable switch. So a bistable switch can decode a gradient, and then that can, in return, that bistable switch can trigger the dynamics of our main protein, which is rho GTP, via an excitable dynamics process. So in practice, when you start combining this type of modules together, you're actually bringing two main paradigms in the field of protein pattern formation. One is, of course, Turing's idea, where proteins self-organize from a homogeneous state into patterns via reaction and diffusion, so via combining bistability and excitability. And then another one is Wolpert's idea, where the cells have a notion of position via gradients. Now, when you start combining these modules together, effectively what a cell is doing is performing computational operation. So on the left is really a schematic of what we thought is happening in the system, and on the right is the actual data. So the way to read this is basically in the first row, what I'm showing you is that how basically this one of this bistable switch is measuring a threshold. When you combine gradients and bistability, you're measuring a threshold. And then in the second row, what I'm showing you is that how you can take then a temporal derivative to basically pinpoint where this mechanochemical response should occur, which then leads to the desired function, which is a certain cell shape deformation and ultimately cell division. Now, as I mentioned, one of the our goals is to be able to write down the theoretical description or mathematical equations that describe the system. And when we do that, it actually gives us the opportunity to do many other things. So when you have a set of partial differential equations, you can now start playing this game both in silico, but also in the lab. So in silico, you can start changing the parameters and see what happens to the dynamics of the system. And in fact, you can now, because our models have predictive power, we can go and exactly do the same thing in experiment. So let me show you what we did now that we know exactly what what are the mathematical equations describing this system. So based on our theory, if you can overexpress our bistable switch instead of having just basically one contraction wave going along the cell, you will end up with many of these. Basically, you end up in a local oscillatory dynamic, which means that every part in the cell is now going to get excited. So you should, instead of seeing just one wave propagating, you should see many of these waves propagating. And in fact, that's the case. So let me show you one example. So you can change experimentally the parameter, the bistable parameter in the system. And now instead of having just a traveling wave, you have a traveling wave with all these spirals that comes behind it. And this is just the beginning, because you can now, what we usually love to have in physics is a control parameter, which we can fine tune this control parameter and basically get the desired response that we have from the system. And this is an example of that. Again, I'm changing the bistable switch, the concentration of my bistable switch in the system, and I can generate a kaleidoscope of many of these patterns in a living system. Each of these patterns, you can see that they change the fluorescence intensity. So the patterns that you are seeing is this protein and its concentration on the membrane. So you can see that they have usually different oscillatory dynamics. They have different wave patterns, wavelengths. And this basically gives us the opportunity to learn a lot more about this system. And I'm going to show you one thing that we have learned about this system. So as I mentioned in my introduction of the talk, we would like to have basically a physics language to be able to describe what we see in the system, not different from other systems in physics. And so the way we define order parameter in a system like this is we're going to play this game of just looking at one point on this membrane, and then we're looking at the fluorescence intensity signal on this point. And if you plot it in time, you can see that it shows an oscillatory behavior. So what you can do is, instead of looking at the magnitude of the oscillation, you can look at the phase of the oscillation. And then that transforms this boring black and white video that you see into this colorful video of the relative phases of the oscillatory signal across the membrane. And it turns out that this phase field actually has a lot of interesting information. So I'm going to show you one example of what kind of information you can gain from this. And I've been having a lot of fun with this, but let's look at this phase field. And one thing you will notice is that there are points on this kind of field, or basically this phase field on the membrane, where the phase is not defined. So you can see that you have points that are now here colored with blue and red, and that's where the phase itself is not defined. So we call that a singular point. When you can see that when we go around a singular point, you can either have a clockwise rotating field or a counterclockwise rotating field. And based on the direction of the rotation of the field, you can assign a charge to these singular points. And here you can assign a +1 or -1. I'm not going to go into details of it, but this is the stable charges of +1 and -1. And now what happens is that I have reduced the complexity of a living system in its glory to actually a cloud of +1, -1 charge, which actually behaves exactly how you would expect. So you can have a fluctuation in the field, which results in a creation side, and you can have +1 and -1 charge that annihilate each other. And again, this is basically experimental data of a very complex system as we know it. And we've been having a lot of fun with this. So for those of you in the audience who are interested in what kind of physics you can do with this, you can look at, you can show that it behaves essentially like a turbulent regime. And other thing you can do is you can take these trajectories and now you can expand those trajectories in time. So this is basically showing a slice in XY and then a slice in time. And now you can see that these trajectories, they become word lines when you include the time axis. And then you can start playing this game of looking at how they braid with respect to each other. And this is kind of like ideas that a lot of people in quantum information processing has been thinking about, how we can encode or decode information by basically braiding these type of charges. And I will leave it at that and end this part of my talk by just saying what's, I think, more exciting to us is the fact that once you learn how biology does it, you can actually, and not be afraid of the complexity of it, now you can try to tame it. And this is what we've been doing. So far, we have played with one control parameter, which we can change and generate a range of interesting phenomena in the system. We can even do more than that. So on the left, you can see this video, which is an in silico demonstration of these waves. And then you can see my cursor is appearing. And when my cursor is appearing, I'm locally changing the reaction and diffusion terms in the system. So basically, it's a simulation. So I'm locally changing that. And now you can see that I can locally generate a defect and then try to braid that defect by moving my cursor around. And how do I do this in experiments? I can use light. So I can engineer proteins that have a light activation, a light activable domain. Now I can recruit the proteins wherever they want. So I can do all these games of actually changing the shape of the cell, however I want. So I can create, for instance, this square shaped cell. And again, we can learn a lot more about how information is being encoded and decoded when you have patterns of like this in the cell. Okay, so I'm going to end this part of my talk and then quickly move on to the last part, which is something I'm very excited about. I'm just going to show you a few interesting observations when we go even beyond the cellular scale. So now we're going to at the organismal scale. And I'll show you what we discovered, this new material properties that can have very important implications for material design. And again, this is how physics can give us this bio-inspired approach to learning about these systems and then trying to implement it. And to do so, now we're going to take one of these egg cells and we are going to fertilize it. So in this first frame, you can see that the fertilization has begun, and you can see that the cell division, now symmetric cell division is happening. So you get first symmetric cell division. This is followed by many, many events. So I'm going to try to speed up this video if I can. Guess not, but you can see that this process is happening. And while this process is happening, you can see a lot of rearrangement within this fertilization envelope. So a lot of cells, millions and millions of cells are being reorganized. And what happens is that you can see that at some point, these cells try to, these embryos, so they're beyond a cell, they're millions of cells, they're trying to kind of like oscillate within the fertilization envelope. And this is because there's a group of cells that's sitting at the periphery, and they have a very important organ known as cilia. And cilia is a hair-like protrusion that beats, and it's responsible for swimming and many other events inside these living organisms. So this is a close-up of one of this. So you can see that each of these embryos are now covered with these cilia. And then if you look at closer, you can see that the cilia is beating, and it has a certain beating pattern. So it basically looks like metachronal waves that you might see in a stadium. And then you can see that now when these beats, they result in each of these embryos being able to swim and start looking for food, basically for further developmental processes. Now, something very interesting happened when we put a large number of them together. And just to orient ourselves, each of these small spheres are one of these embryos. We put a lot of them together, and then we're going to watch what happens. And they say a picture speaks 1,000 words, and this is a video, so I will let you do the math. And what you can see is basically we have a large crystallization of basically a big crystallization of a large assemblies of these starfish embryos. that they come together over the course of their development. So initially they form a crystal, and then after a while the crystal fully dissolves. And this is a process that happens over many days. It's also a process that you can observe with eye. So one thing to note here is that there is a certain rotation that corresponds to this, and then we're going to learn where this rotation comes from and actually what are the implications of this. So what I'm going to do is maybe first to give you an idea of the scale. This is my shot on iPhone image, and you can see that this is actually looks like glass beads that are sitting at a water interface. So why does this happen? So I'm going to give you an intuition about why this happens. So first of all, here you can see one of these embryos, and you can see that it's slightly elongated along an axis of symmetry, which is called an anterior-posterior axis. And when the embryo swims towards the water-air interface, it actually can attain a stable configuration such that their AP axis aligns perpendicular to the interface. And here you can see the fluid flow around it. You can see that it acts like a dipole. So now when you have one of these at the water interface, because of the fluid flow around it, it can attract other stable embryos at the surface. And this results in the formation of a small nuclei, which later on grows to a larger crystal. And one thing to note is that as the development progresses, you can see that, first of all, the shape of the embryo becomes more and more elongated, but the fluid flow, which is this dipolar view, basically this dipole around it, the strength as well as the magnitude of it is changing with time as well. So which that essentially results in final dissolution. It becomes weaker and weaker, and it results in a final dissolution, dissolution of the crystal. So on its own, it was an interesting phenomenon. But what was really unusual about this was the fact that there is, if you think about a crystal, when you have basically a crystal within a certain, you have atoms within a certain crystal. The atoms, they have a certain reciprocal attractive force between them, which affects their potential energy, which if you remember, the potential energy is the stored energy that's determined solely by the relative position in the crystal. Now, something unusual about this crystal is the fact that each atom within this crystal also has a rotation with respect to its neighbor. They all have a certain handedness, which is because of the beating of the cilia. So this additional rotation creates an additional force and torque exchange in the system, which is non-reciprocal. So it defies Newton's third law. And in a minute, I'll show you what's the consequence of having non-reciprocal type interactions, which actually is a result of the non-equilibrium nature of this living matter. So again, just going back to everything we did, we can write down equations which can, so this is a simulation of the process because we can identify all the important variables and constrain them using the experiments that we do. So I'm going to skip this and then show you what happens when you have this type of force and torque exchange in the system. So if you remember, we have a global rotation of the crystal. And that's, of course, because each of these atoms are rotating. But then if you go to the co-rotating frame of the crystal, so if you go and sit on the crystal, what we observed was something very unusual, which is the fact that this crystal has sustained oscillations. And this should come as a total surprise because this is an overdamped system. This is a system that's sitting in water. So all the oscillations should damp out very quickly. Yet this is a system that can basically sustain this type of oscillations for very long periods of time. And so I'm going to I'm going to skip this. And again, because what we discovered was that because this system, you don't have energy conservation, the golden rule of any equilibrium system, your elastic, it's basically your elasticity tensor will have new additional terms, which is called odd elasticity. These are off diagonal terms. These are technical terms, so I'm just going to gloss over. But what it means is that this is a system where you can generate work by not continuously putting energy in it, but just by kicking the system once, and then you can actually generate an engine. And this is an example of how this works. So for instance, we can put this system and now have a material that's doing work on its environment. So it's unlike any other engine that we have learned and know to love, for instance, in thermodynamics. So I'm going to, I know I've gone maybe over time, so I'm just going to wrap up my talk by maybe leaving you with 2 points. One is the fact that what's I think the natural world give us this opportunity to not only try to account for the phenomena that we observe, but it also allows us to do new physics, to not only make sense of life, but to also enrich physics itself. And I think this is why we're really at the next frontier of what we can do in physical systems. And every time, I think every time we have a new class of phenomena, it requires new concepts, which actually ultimately results on formulation of new laws. And living systems is really now this incredible opportunity to push the boundaries of physics in ways that was not possible before. And this is for two, I guess, articles. One is a perspective, for more technical perspective that I've written with my colleagues. And the other one is an interview that I did, which also highlights some of the work that we have been doing in my group. And I'm going to finish my talk by just showing you the wonderful, amazing people in my group who are leading this work, my graduate students, the undergraduate students. This list is actually very long. So these are the ones that have been more involved in the work that I described today and are also a part of the group now, the postdocs that have been a part of my group, and my colleagues both at MIT and across the globe. And I'm happy to take questions.
Thank you, Professor Fakhri. I don't know if I should characterize you as enthusiastically articulate or articulately enthusiastic, but I am overwhelmed by the energy and the capacity. One learns in science, at least from the physics perspective, that all of science is physics, just at different levels of complication. And you certainly seem to be proving that at a level that many of us have never seen before. We do have a number of questions. I'll try to get to as many as I can. I have seen, Andy asks, I have seen lectures on recognizing learning in a very simple sea snail. At the other extreme, neurologists discussing neuromotor control in Parkinson's disease and arguing that on or off behavior depends on system imbalances and strands connecting the basal ganglia to the cerebellum, as opposed to other scientists who insist that the explanation of Parkinson's is genetics. You're welcome to respond.
I mean, I think that's always maybe within the biologists, that's also kind of like a question whether everything depends on genes or one. I think we're moving beyond that, and there certainly are a combination of. I mean, of course, you have a genetic makeup, but then of course, the environmental and everything else around you will also dictate the type of behavior and everything you see. I would say that the physics of behavior is potentially another level of so I talked about complex systems, but I think going to behavior, that's even more complex. And some of the ideas that I described today is something that people, when they think about behavior, they're also trying to understand is where they're like keywords such as non-reciprocity or kind of like this maybe irreversibility, or all these features can be used to describe like a collection of neurons all the way to the brain, all the way to the behavior. And I think that's one of the most interesting and exciting directions to explore in terms of complexity in complex systems.
Thank you. Robert asks, does possible liquid crystallinity of DNA and proteins in the concentrated state play a role in cellular processes?
I think also a very interesting question. In fact, within the physics department, there are many people who think about these questions. One of my colleagues, Leonid Mirny, he thinks about how you can kind of like get this 2 meters of DNA and then kind of put it into like a micron size container, but yet like really have processes kind of like happening exactly where they should happen. So it's much smaller scale. And I think he uses ideas from polymer physics to really try to understand and go beyond just, for instance, genetics, or beyond like basically use physics to try to explain this phenomenon.
Thank you. Winston asks, can the correlation of about 1 second in the infrared fluorescence emission from actin-myosin network be due to diffusion of an emitter to a place where another emitter was a correlation time earlier?
I guess I will maybe, interesting question. So first of all, we're not labeling actin or myosin. So these are granules that are sitting within an actomyosin network, and the fluorescence comes from this granules. I would love to have a chat later, like about we're trying to identify what it is. It turns out it's probably a protein and cofactor, and they're not close enough. So if you think about it, I mean, what I showed is a 200 micron field of view, and then they're within like a few microns of each other. So there's no way of like kind of having any energy transfer between them. Yet within each of these granules, we probably have a lot of interesting photophysics, because I would assume, think that you have a lot of fluorophores within one of these granules, and probably there's a lot of interesting energy transfer within one of this. So the photophysics of this is, again, maybe a different talk, but it's very interesting on its own to try to explore.
Another audience member asks, more on a personal note, when did you become interested in science and scientific research? What factor or factors generated that interest?
Yeah, I guess it's a bit of a cliche to say that I always wanted to be a scientist. I'm very fortunate to have parents that were very encouraging from very early on. So yes, I had my chemistry kit at a microscope very early on. I did go into engineering, and that was more of a family, I guess, push for me to become an engineer. But I think I was always a closeted physicist. And then when I had the opportunity to kind of like become a physicist, I just went for it, and I can't be happier.
Good. And one more, I believe. Andy asks, please excuse this armchair engineer trying to link your talk to the concept matter behavior for inert of the simplest living cells to genomes to neuron to neural strands to complexity, increasing all the way to oscillating systems in the brain. I'll let you reflect on that.
Okay, yeah, I mean, again, I guess I'm happy to chat more about this. So I do think that complex systems has been something that was like, there has been this disconnect between how physicists think about systems. They always loved simple systems, and of course, engineers, they think about complex systems at all times. But I feel like when you start thinking about living systems which are inherently complex, then you need to bridge this. And so I think this is kind of like a wonderful opportunity. And like, it's not just living systems. I think even climate, like the physics, that the systems that we care about today are complex, and the science of complexity, I think, is where we can make progress. And so not to be afraid of the complexity, but try to tame it, whether it's living systems, try to understand what life is, what life life on exoplanet means, or what climate, how do we control climate? I think this is all things that hopefully can be answered by physics, and of course, boundaries between other fields.
One more, and then we're going to have to wrap up. Larry asks, is the orientation of the AP axis independent of gravity?
They're actually they are actually heavy particles, so they want to sink, and the fact that they rotate keeps them kind of like at the water face in. So if you lightly remove the cilia, you can see that they sink to the bottom. So there's kind of like certainly some very interesting, I guess, things to think about, or just try when you think about material design in general.
Thank you. I think we're going to call that a wrap. As I said, your articulate enthusiasm is exceeded only by your enthusiastic articulation. I very much appreciate your contribution and your time you spent with us today. I'm now going to bring this exercise to a close. Thank you, ladies and gentlemen, for participating. Thank you, Professor Fakhri.
Thank you very much for having me. This was wonderful.